Hello everyone, my name is Rebecca Boys and I'm a PhD student at Massey University in New Zealand. And today I'll talk about part of my PhD project, which is investigating the ethology of stranded cetaceans. So New Zealand has an international reputation for its high incidence of strandings and its extensive public engagement in strandings response. Yet in many cases, these procedures have not been validated by empirical data, and there remains a lack of robust science to inform these interventions, including for evaluating animal condition. Now, importantly, the implications of these human interventions on the welfare and subsequent survival of cetaceans remains unknown. Unfortunately, there's no currently systematically collected data on the behaviour of stranded cetaceans, which are in atypical, stressful situations. And this limits our ability to assess animals and examine the effects of human intervention to assist in decision-making for stranding's response. However, since stranded cetaceans are open to a variety of stresses, it is likely that they do elicit a number of indicators which, if understood, could be used to assess their welfare. But this first requires the identification of practically measurable and observable indicators and their monitoring at a range of strandings. So to collect this data, I'm attending stranding events around New Zealand. But today I'll focus on data collected from a mass stranding of 2 pygmy killer whales that occurred in the north of New Zealand in 2020. And both of these individuals had re-stranded. So whilst undergoing individual assessments, each animal was filmed using 2 GoPro Hero 7 cameras to examine the behavioural events and some physiological events, such as respiration rate. These cameras were mounted 1 to 2 meters from each side of the head of the animal and faced down the body towards the tail flukes, enabling the entire body to be observed throughout the stranding. Now, the video footage that was collected was then examined and coded using program Boris. And this data was coded using an ethogram, which I built a priori based on some information in the literature, and which was then updated based on the behavioural and physiological events that were displayed during the stranding event. I also classified the human interventions and coded these in program Boris, and this data was used to quantitatively examine human intervention and assess whether there were differences in behavioural events displayed by stranded cetaceans, using the duration of these behavioural events with and without humans. So the first individual was filmed for 3.5 hours and the second for 1.5 hours. And from the video footage, we classified 19 different behavioural and physiological events. 18 of these were displayed by the first animal and 12 by the second. But the number, frequency and duration of events did differ both between the individuals and also over time. So these 19 different indicators were broken down into categories based on the area of the body involved. This provided us with whole body movements, those that involved only the head or tail, those related to the blowhole, and also related to the fins. So this graph shows all 5,474 events that the first individual displayed, and these have been split into those 18 different indicators. And you can see that this individual was displaying movements over the entire stranding, with more than 97% of total stranding time displaying this tail fluttering. Now, to be able to examine changes over the course of the stranding event, the data was split into 30-minute periods. And we can see on this graph those measured as a frequency of occurrence. We see that respiration generally shows an increase over time, whereas the blowhole twitch and the lower jaw movements both decreased over time for the first individual. So the head movements were measured as a proportion of stranding time based on their duration. And we can see that the head side to side generally showed an increasing trend, whilst head arching decreased after the first 30 minutes of stranding and then remained relatively stable. The head lifting, on the other hand, showed a decrease until halfway through the stranding and then began to increase again. Tail and fin movements were also measured as a duration. So we see that both tail arching and tail side to side generally showed a decreasing trend, but the dorsal fin flutter showed quite a sharp increase until partway through the stranding and then rapidly decreased. The second individual was much more lethargic, showing far fewer events. The event displayed most was this head side to side, but this still only accounted for 26% of stranding time. And this individual did display one event not seen in the first individual, which was the opening and closing of the mouth. Again, the data was split into these 30-minute time periods. And we can see that the respiration and blowhole twitch for this animal showed generally a decreasing trend until halfway through the stranding and then increased slightly, whereas the lower jaw movements showed a rapid increase until halfway through the stranding and then rapidly decreased. The head and fin movements were again measured as a duration. And we can see that the head lifting and the head side to side both showed an increasing trend, whereas the dorsal fin flutter showed a decreasing trend. And this is similar to that was observed in the first animal. Tail events were also measured as a proportion of stranding time. So the tail lift remained relatively stable over time, whilst tail flutter remained relatively stable until halfway through the stranding and then showed this rapid increase. And the tail hover increased slightly until partway through the stranding and then decreased. So we also classified the human intervention that occurred, and this was considered as any time that a human or dog was within 2 meters of the animal. Now, the manipulations were classified into 6 different types: noise, digging, reflex testing, touching, rolling, and watering. And all of these events occurred with both animals. Now, for both animals, the presence of humans was almost continuous throughout the stranding, whereas the manipulations varied. So the digging, rolling, and touching of animals generally decreased over time, whereas watering tended to increase. We assessed differences in the behavioural and physiological events displayed by the animal during human intervention by comparing the proportion of event duration. And we found that when there was human noise other than normal talking, when humans were rolling or digging around the animals, or when a dog was present, that the events head side to side, respiration, and dorsal fin flutter all showed an increase in their duration. So this study provided the first data on the ethology of stranded cetaceans and allowed us to quantitatively examine human intervention. Overall, this enabled us to build the first stranding-specific ethogram informed by quantitative data, which can now be used to examine events at future strandings. So I'd really like to thank you all for listening and thank our funders and collaborators for their continued support. And please do feel free to email me if you have any questions.